The chemistry of life. In AP biology, you'll focus on four main macromolecules: carbohydrates, lipids, proteins, and nucleic acids, otherwise known as what you'll typically find in bread, fat, meat, and DNA. These macromolecules are called polymers, or large molecules made up of smaller subunits. These subunits are known as monomers. All these macromolecules are polymers except for one type, lipids, and we'll look at that in a minute. Monomers are held together by covalent bonds, and each of the four macromolecules have their own type of covalent bond. When a monomer and a monomer join to form a polymer, this process is called dehydration synthesis. The reverse reaction is when a polymer breaks down into monomers, and this is called hydrolysis. Dehydration synthesis and hydrolysis both have something in common. They both have something to do with water. In dehydration synthesis, monomers lose water to join together and form a polymer, and in hydrolysis, when water is added to a polymer, it breaks back down into the monomers it was made of. A good example of dehydration synthesis would be the production of maltose, which is a polymer made from two glucoses, or two individual monomers. As you can see, the hydroxide group from one glucose and the hydrogen from the other glucose make up the two hydrogens and an oxygen found in H2O, or water. When the H2O leaves, it leaves an opening on both of the glucoses to join together and form maltose, and water is a byproduct of the reaction. This is why it's called dehydration synthesis. The molecules needed to build a water come out from the monomers, become a water, and this allows the monomers to join and form a polymer. Here's an example of hydrolysis. This is a disaccharide, or a polymer, and we'll look more into disaccharides later. When water is added, the water molecules split into that OH, or the hydroxyl group, and the H, and this breaks apart the polymer by binding to the polymer where the monomers would be joined together, thus breaking apart the polymer back into monomers. Now let's look at the four main macromolecules of life. Let's start with carbohydrates. Carbohydrates are made from the elements carbon, hydrogen, and oxygen, or CHO. Carbohydrate monomers are called monosaccharides, mono meaning single and saccharide meaning sugar, so a monosaccharide means single sugar. Polymers formed from these monosaccharides are called disaccharides if they are made up of two monosaccharides, like maltose. If there are more than two monomers involved, then it's called a polysaccharide, meaning many sugars. Bonds between carbohydrate monomers are called glycosidic bonds. Carbohydrates serve many purposes in life, one of them being an immediate energy source. Glucose is a monosaccharide that is one such immediate energy source, and it can be used for ATP production. Cellulose is a polysaccharide found in plant cell walls, and cellulose serves to keep the plant upright in the absence of a skeletal structure. Because plants don't have bones, they rely on the cellulose in their plant cell walls to keep them upright. Glycogen is another polysaccharide, and glycogen stores excess energy or glucose in animals and fungi. Glycogen is typically found in the liver of most animals. The next macromolecule we'll look at is lipids. Lipids are made from carbon, hydrogen, oxygen, and sometimes phosphorus, and this sometimes we'll look back in a minute. In the beginning, we mentioned that lipids aren't considered true polymers, and here's why. The subunits found in lipids aren't necessarily directly bonded to one another, so lipids don't technically form polymers. However, they still have subunits, like fatty acids. These just don't bond together in a way that justifies them being called proper polymers. There are two main subgroups of lipids you should know about for AP Bio: saturated fatty acids and unsaturated fatty acids. This is a saturated fatty acid. You can remember the structure of saturated fats by keeping SSS in mind. Saturated fats have single bonds and are solid at room temperature. So notice how all the carbons are held together by single bonds. This allows for less fluidity, as single bonds are more rigid than double bonds. This is why saturated fats are often solid at room temperature, like butter, due to these rigid single bonds. This is an unsaturated fat. You can see that there's a kink in the structure, and this is caused by a double bond, something not found in saturated fats. This double bond allows the molecules to move more freely, so unsaturated fats are technically more found as liquids at room temperatures, like oil. We mentioned how lipids sometimes have phosphorus. When this happens, the lipid is called a phospholipid. This is a cup of oil and water, and you can see that they don't mix. They don't mix because water molecules are polar, meaning they have an electrical charge. Lipids, for the most part, are nonpolar, meaning that the molecules have no electrical charge. Water will only associate with other polar molecules, and which is why we see the separation of oil and water here, because water only wants anything to do with other molecules with a charge. Lipids having no charge won't mix with water. However, with phospholipids, the molecules have both a polar and a nonpolar region, meaning they are both hydrophilic, water-loving, and hydrophobic, water-fearing. The phosphate head is polar, and that's the part that likes water, and the fatty acid tails are nonpolar, which makes them hate water. Now this is a cell membrane which is made up of a double layer of these phospholipids, which is why it's called a phospholipid bilayer. Since the outside of the cell is watery, meaning polar, the likewise polar parts of the phospholipid, or the head, face the outside of the bilayer. The nonpolar tails of the phospholipid want nothing to do with the outside fluid, so they face each other on the inside. Like carbohydrates, lipids play an important role in life. Lipids act as a source of stored energy for the body. They can also protect and insulate neurons in the form of myelin sheaths. Myelin sheaths are fatty substances that coat neuron bodies, and these insulate electrical impulses in your brain and help them go faster. Lipids also act as cell membranes, as we saw previously, and are also the building blocks for several hormones. When a hormone is made of lipids, it can easily pass through a cell membrane because the phospholipid bilayer of the cell membrane and the hormone are both nonpolar, so the cell will easily allow the molecule to pass through. Next, let's look at proteins, which are made of the elements carbon, hydrogen, oxygen, and nitrogen. Protein monomers are called amino acids, and their polymers are called either a dipeptide if there are only 2 amino acids, or if there are more than 2, then they're called polypeptides. The bonds between the amino acids are called peptide bonds. This is an amino acid. On the right we have the carboxyl group, which is the part of the amino acid containing the oxygens. On the left we have the amine group, which is where you'll find the nitrogen. At the top we have a hydrogen, and at the bottom we have the R group, or the variant group, or the side chain. This side chain is what determines the protein's structure and function. It is different for different types of proteins. For AB Bio, you'll need to be familiar with the 4 levels of protein structure, or essentially how amino acids come together and fold into proteins. You have the primary, secondary, tertiary, and quaternary structures. Now for the primary structure, this is the peptide bonds between each amino acid in a polypeptide chain. The secondary structure consists of the hydrogen bonds between the atoms of the backbone of this polypeptide chain. These hydrogen bonds either cause the chain to fold into a beta-pleated sheet or into an alpha helical shape. The tertiary structure is the folding caused by the R groups, or the side chains mentioned earlier. For example, polar side chains will join with other polar side chains, and sulfur-containing R groups will join with other sulfurous R groups to form disulfide bridges. Here you can see those alpha helices and the beta-pleated sheets we mentioned previously. The quaternary structure of a protein is the orientation and arrangement of multiple tertiary structures. So if a tertiary structure was a cake, then the quaternary structure would be the multiple cakes arranged together. In protein, structure determines function. A structurally damaged protein is called a denatured protein, and this can be caused by high temperatures or a suboptimal pH. Proteins are important to almost all aspects of life, especially in the cell membrane. The blue structures here you see are all proteins, and they serve as channels for molecule transport, cell-to-cell communication, and overall membrane stability, as well as they do much more in cell functioning. Next we have nucleic acids, which are made from the elements carbon, hydrogen, oxygen, nitrogen, and phosphorus. Nucleic acid monomers are called nucleotides, and their polymers are called dinucleotides if there are 2, and for polymers of over 2 nucleotides, then the polymers are called polynucleotides. There are many different types of bond types found in nucleic acids, and all you'll need to know is all the monomers are covalently bonded. This is a nucleotide. Nucleotides are made from a 5-carbon sugar, which is either deoxyribose if it's DNA, or ribose in RNA. Attached to this sugar is a phosphate group, which together with the sugar creates the sugar-phosphate backbone of DNA and RNA. Next, you have the nitrogenous base, which would be one of the A's, U's, G's, C's, and T's we see in DNA replication. We know that nucleic acids create DNA and RNA, which is something that follows the rule of directionality, or the direction in which the DNA or RNA strands run. You won't need to know the details, but you need to know that DNA is anti-parallel, meaning the two strands run in opposite directions because the nitrogenous bases have to align. The two strands have to complement each other with the corresponding bases, cytosine with guanine, adenine with thymine, etc. DNA runs from 5' to 3', and these numbers just refer to the carbons on the 5-carbon sugar. You won't need to know the details. If one strand of DNA from top to bottom is 5' to 3', then the other strand from top to bottom must be 3' to 5'. Again, AP Bio does not delve into the details of carbon-sugar alignments, but it's good to know the general directionality of DNA, which we'll look more into for DNA transcription, translation, and replication, but that's for another video. Just to recap, carbon is a fundamental block of life and is found in all organic matter, meaning all life on Earth contains carbons in one form or another. Next, you should know that water controls the breaking and forming of polymers. You add water to break up a polymer in hydrolysis, and you remove water from polymers in dehydration synthesis. To finish up, the four main macromolecules are carbs, lipids, proteins, and nucleic acids, which are all polymers made of subunits called monomers except for lipids. We hope this was useful, and we thank you for watching.